Okay, I'm a postdoc in Hazel Siv's lab at the Whitehead Institute, and it's my pleasure to introduce our next speaker, which is my peer at work, Jasmin McCammon. She did her graduate work at Berkeley and joined our lab about 3 years ago. Her background was in zebrafish, and she just hit the ground running. She's been great to work with. She always has new ideas and things to add to our project. So I'm going to let her tell you about the interaction and synergy studies that we've been doing in 16p11.2.
Okay, thanks, Alicia. Thanks, Alicia. Alicia could just as easily be giving this talk because everything I'm going to be talking about today we've been working on together, but I'm really excited to tell you about it. Okay, so there are a lot of components that contribute to making understanding and treating autism an uphill battle, and I think achieving these ultimate goals will really require embracing complementary and different approaches and really utilizing the strengths of these different approaches to attack these different components. And I'm here to tell you about our approach, which is to use the zebrafish as a uniquely suited system for rapid genetic analysis to hopefully complement the other great approaches we've heard about today. And then I'll tell you our studies on the 16p11.2 CNV genes. Okay, so animal models are very important to understanding clinical impairments, and they also play a big role in developing safe and effective therapeutics. And what I want to emphasize is that it's good to work with multiple animal systems, because then you can actually end up diversifying the feedback you get through these systems, and that makes our work more efficient and more effective. And the easiest way to manipulate these different animal systems is to change their genes. But what isn't easy is the complexity of the genes involved and the fact that they're likely to interact with one another. So this is a partial list of genes from the MindSpec database that are associated with autism. And there are genes associated with syndromic forms and rare variants and common variants and candidates from genome-wide association studies. And I think this is a snapshot of the complexity we're dealing with in this post-genomic era of personalized medicine. And the way we're choosing to tackle this is to use the zebrafish. And the zebrafish is actually really great because it can take a large data set like this and sort of act as a funnel or to filter out priority candidates that can be followed up on in more complex systems. So how does the zebrafish do this filtering? Well, first, I would like to emphasize that we prefer the term tool over model because zebrafish cannot get autism, but they do have conserved genes and pathways, which implies that they can facilitate the understanding of the basic underlying biological phenomena. The adults are small, and they're easy to maintain, and a single mating pair can give up to 200 embryos at a time. These embryos are easily accessible because the fertilization is external, and they're actually transparent, which is really nice for imaging deep tissues, such as the brain. The zebrafish genome is fully sequenced and well annotated, and in fact, there are, for the 84% of human disease genes in the OMIM database have a zebrafish homolog. There are a wide variety of genetic and molecular tools at our disposal for rapid gain and function analyses. And finally, because the embryos are small, we can actually array them out on a plate and use them as a whole organism readout for the effects of large chemical libraries. Okay, so to show you a zebrafish embryo in action, I have a time-lapse movie. It starts when the embryo is about 6 hours old, so it's composed of several thousand cells that have migrated down to cover this big yolk ball. And as development continues, the embryo expands. You can see the eye is going to start to form here. Muscle segments are being added sequentially from the anterior to the posterior, and this tissue extends into the tail and goes away from the yolk. The central nervous system is developing, and in this lateral view, you can kind of see that the ventricles are swelling with fluid. And these dark spots here are actually blood cells that are flowing because the heart has started beating. So basically, by 24 hours, this embryo has formed all of its major organs. So we're using this speedy zebrafish to study another genetic aspect, which is copy number variants. We've heard some about them today, and they're highly associated with autism. But moreover, they represent a sort of way that we can study complex genetic interactions in a more manageable way, because they represent a more limited set of genes as opposed to that original slide I showed you. So you guys know now the copy number variants are either deletions or duplications of a region in the genome that results in a change in the copy number of genes in the affected region. And perhaps unsurprisingly, it's been shown that there's a correlation between the number of genes in the CNV and the disease burden. And one hypothesis then is that changing the dosage of one or more of these genes is what links the CNV with the disorder. So therefore, it's absolutely critical that we begin identifying the pivotal genes from these regions so that we can know their mechanisms and get to understanding pathology. So one CNV that's consistently cropped up is the 16p11.2 CNV. This is a graphic from a review last year that shows some common recurrent CNVs. And I'll draw your attention to the blue bars, which are the incidents associated with autism and these CNVs. And you can see that both the deletion and duplication have more cases of autism associated with them compared to these other CNVs. And together, they account for about 1% of autism cases, which is actually really significant when you think about the overall incidence of autism and compared to other genetic correlations. But the CNV has 25 genes in its core region, and the critical ones are not known. So to get at finding these critical genes, we can begin by looking at single loss of function studies. And in Zebrafish, this is really pretty fast and easy. So the day before, you set up the male and female adults in mating tanks, and the adults are triggered to spawn by the light cycle. So the next morning, you'll have hundreds of single-celled embryos that you can easily inject with antisense oligonucleotide morpholinos, or MOs. MOs are these short oligonucleotides that can be designed to target the mRNA from your gene of interest. And when it binds by base pair complementarity, it will either inhibit its translation or splicing, so that results in no or defective protein. Now these embryos have formed, their major organs by 24 hours, so you can begin to look at early loss of function phenotypes the next day. So we've previously shown that of the 25 core genes, there are 22 homologs in zebrafish, and of these, loss of function in 20 of them results in altered brain morphology. So what do I mean by this? Well, this is a picture of our control morpholino-injected embryo at 24 hours of age. It's a dorsal view looking down at the head. And we've injected the ventricular space with a red fluorescent dye to help visualize the morphology. So here are the eyes. This is the forebrain, midbrain, and hindbrain regions. And you can see they undergo a characteristic bending and folding, which gives them a stereotypic shape. And here's a subset of our loss of functions. And you can see here that shape is lost in some or all of the regions. We've also shown that the human genes and zebrafish genes are conserved and can function in place of one another. And the way we do that is to induce loss of function in the fish version of the gene, and then we can co-inject the human RNA to see if that can substitute for the loss of the fish version. So this is just one example of HERIP3. Here's our control embryo again. And when you inject the HERIP3 morpholino, you can see we get this abnormal brain morphology and body shape, which is restored when you supply the human HERIP3. Another thing we can do is start to examine which genes might be sensitive to copy number. So we can titrate the amount of morpholino until we're only looking at about 50% loss of function, which we'd assume to be, approximately equivalent to the expression levels in a 16p deletion carrier. So we did this and then we can look to see if the embryos still have their phenotype. So we did this for 14 genes, and I'll show you 3 of them here. So you can see that for ASPHD1, at about 50% normal RNA remaining, this light gray bar means that there's no phenotype. And you, in fact, have to decrease the normal RNA to about 25% of the control levels in order to get a phenotype. However, for aldoa and KF22, this high morpholino dose that gives the phenotype is actually equivalent to about 50% loss of function, implying that these genes are important because they may be sensitive to dosage and to copy number. So because aldoa is a dosage sensor, we wanted to do a small sort of hypothesis-based chemical screen to see if there was a chemical compound that could rescue the aldoa loss of function phenotype. So aldoa A does have these other moonlighting functions, but it's sort of known and defined as one of the enzymes that catalyzes the breakdown of glucose into pyruvate, which produces energy for the cell. So therefore, you would predict that if you lose aldolase A function, you lose pyruvate production, and there's less energy, which may be contributing to these phenotypes. And in fact, if we co-inject pyruvate with aldolase A morpholino, we can actually rescue this narrowed brain morphology and sort of abnormal muscle segments to get embryos that more closely resemble the controlled condition. So these single loss of function experiments are really excited, but we're worried that we're missing something on the interaction level because thousands of patient exomes have now been sequenced and no single variants of statistical significance have been recovered so far. So to me, this implies that there's more than one important gene from this region. And to find that, we could go with candidate gene approach. But this list is somewhat mysterious, and nothing obvious always jumps out, and we'd be worried about missing something. So we chose to use a zebrafish to look at genetic interactions in an unbiased manner. So to do this, we again used our assay to look at 24-hour brain morphology. And we chose this assay because we think it represents sort of a high-level evaluation of central nervous system development and captures a lot of the processes involved therein. So our previous study, we know that with about 50% loss of function, most of the genes don't have a phenotype. But what we're interested in seeing is if they can interact to impact brain development. So a real-life example of this is MAPK3 and SEZ6L2. You can see the single loss of function embryos resemble the control morpholino injected embryo, whereas when you put in the same amount of morpholinos together, this results in a dramatically narrowed tube and the short and curved body axis. And you can score the different phenotypic classes and see that this abnormal brain is very penetrant in the double loss of function condition. So we tested MAPK3 against all the 16P genes. And we only saw an interaction with SEZ6L2. But in order to do all of the pairwise interactions, you actually get a massive number of things to test, which is a pretty challenging in vivo problem, but something that rapid zebrafish assays are well suited for. So we tested all possible pairwise combinations that we could, and we found that 9% of them interacted to give a brain phenotype. I don't really expect you to be able to read this table. I just want you to focus on the red and green boxes, which represent the 15 out of 162 pairs tested that interacted to give a brain phenotype with varying levels of penetrance. And I'll blow up a portion of it so you can see it better. And the 9% is exciting because it shows the majority of the pairs didn't interact. So our assay has some specificity. But 9% is also a lot more than you would expect by chance. If you look at synthetic lethal studies, they would suggest that a random pair of genes has a 0.5% chance of interacting. So you can make a visual representation of these interactions by drawing lines between interacting genes and scaling the font size to reveal these sort of integral hub genes that have multiple interaction partners. And what's really exciting about all these interactions is that they're all completely novel, and none of them have been previously published on. and they wouldn't have been predicted based on what we know about their gene function. So the zebrafish has contributed this new base of information, and we've used it now to prioritize candidates for mutant generation. So mutants, while a lot slower than morpholinos, offer the advantage of being able to study later phenotypes in larvae and adults that you can't use the transient morpholinos for. So the highlighted ones we made mutants for, and I'm just going to tell you about a single gene for one of them, for ASPHD1. So this is our curiosity assay. Zebrafish prefer to swim in these loose groupings called shoals. So we can put a stimulus shoal behind a divider and then put a test fish in to see how much time it spends in the half of the tank next to the shoal versus the other half. So this ASPHD1 heterozygote is very interested in these other fish and spends almost the entire 10-minute trial in this half of the tank. Conversely, its homozygote mutant sibling spends a lot more time wandering the entire length of the tank and doesn't necessarily exhibit a bias to being close to the stimulus shoal. So this is really intriguing, and we'd like to understand the circuits underlying this behavior and see also if genetic interactions can impact this. So in summary, we can use the fish to filter large data sets for priority candidates, and this works because human and fish genes have conserved function. The 16p11.2 and CNVS are among the most highly associated CNVs with autism, but the critical genes are unknown. And our single loss of function analyses revealed some dosage-sensitive candidates that we can follow up on with small chemical screens. And our double loss of function studies showed some really novel interactions for this region that weren't known before. So I want to thank Hazel for pioneering this notion of the zebrafish as a tool and her guidance, and Alicia, of course, for my partner in crime, and the rest of Team 16P and our funding. Thanks.